that we've talked about transcription, the process of copying DNA to make RNA, it's time to turn our attention to translation, the mechanism by which a protein is synthesized following the code contained within the sequence of an mRNA. After Watson and Crick published their structure for DNA, which made the molecular mechanism of inheritance pretty clear, scientists set out to do the painstaking work of figuring out how a genetic sequence encoded in nucleotides is decoded by cells to allow expression of that DNA in the form of proteins. 3 scientists shared the Nobel Prize in Physiology in 1968 for their contributions to solving that puzzle, and how mRNA, tRNA, and ribosomes are involved in the process. Robert Hawley was able to isolate tRNA and worked for 10 years to finally decipher the first tRNA sequence and structure. Marshall Nierenberg made the first clear connection between mRNA sequence and amino acid sequence, essentially decoding the first word in the genetic code. And Har Gobind Khorana synthesized mRNAs with different sequences, which took literally years at the time. Now you can do it in less than an hour. But this work was critical for figuring out the rest of the genetic code. In transcription, a copy of a nucleotide sequence is made using base pairing rules to yield a product, RNA, that is also a sequence of nucleotides. The same chemical language is used, nucleotides to nucleotides. In translation, however, we're switching to a new chemical language, amino acids, hence the term translation to describe the process. In transcription, because it relies on complementary base pairing between nucleotides, there's a 1-to-1 correspondence between the two sequences. A pairs with U, G pairs with C, because of their complementary hydrogen bond donors and acceptors. But when we switch to encoding the amino acids in a protein, there's an immediate problem. We use 20 different amino acids to build proteins, but there are only 4 nucleotides used in DNA or RNA. If one nucleotide were used to encode one amino acid, we could only use 4 amino acids to build proteins, so that doesn't work. If we use a sequence of 2 nucleotides to encode an amino acid, that gives us 4 times 4 possibilities, so 16 amino acids. We're getting closer, but that's still not enough. So 3 nucleotides is the minimum set that will give us at least 20 different amino acids. And in fact, that's what the genetic code is based on. Each amino acid in a protein is encoded by a set of 3 nucleotides in the mRNA, which is called a codon. This is a fundamental property of the genetic code. Each amino acid is specified by a triplet of nucleotides in the mRNA, so the genetic code is a triplet code. With 4 nucleotide possibilities at 3 positions, that gives us 64 triplets to work with, more than enough to encode the 20 amino acids we use to build proteins. If you take a look at this standard genetic code table, all possible combinations of 4 nucleotides at 3 codon positions are shown, along with the amino acids they specify using the 3-letter amino acid code. To find any given sequence, read down the left side of the table for the first nucleotide in the codon, across the top for the second nucleotide, and then the third nucleotide is on the right side of the table. For example, if we want to know what amino acid is specified by the codon GAC, find the letter G on the left, the letter A across the top, which narrows it down to this box, then C, which is the second codon down. GAC encodes the amino acid aspartate. As we'll see, there's no punctuation in the genetic code. These triplets will be read by the ribosome in sequence, with no nucleotides functioning as commas or periods separating the codons. This has important implications when we think about mutations that disrupt this sequence, as we'll see in a little bit. 61 of the codons in the standard genetic code encode amino acids, and 3 are used as stop signals to terminate translation. We'll see how they do that in a bit as well. With 61 codons encoding only 20 amino acids, there are more triplets than are needed. That results in the second property of the genetic code that we'll discuss, which is redundancy or degeneracy. If we look at the codons, we can see that with only 2 exceptions, every amino acid has more than one codon that corresponds to it. For example, UUU and UUC both encode the amino acid phenylalanine. Leucine is encoded by any of these 6 codons. You get the idea. The only exceptions are methionine, which is always encoded by the AUG codon, which is also used as a start signal to initiate translation, and the amino acid tryptophan is only specified by the codon UGG. But every other amino acid has more than one codon. Notice, however, that the converse is not true. There's no case where a single codon could specify more than one amino acid. And if we think about it, that makes logical sense. As the ribosome is decoding an mRNA sequence to build a protein, if codons could specify more than one amino acid, how would the ribosome know what amino acid belongs in that position? It wouldn't. So although there's redundancy, an amino acid could be specified by more than one codon sequence, the codons are unambiguous. Each codon encodes one and only one amino acid. The AUG codon, which encodes methionine, is used as a start signal, and these three codons, UAA, UAG, and UGA, are stop signals that terminate translation. But why are these needed? You'd think that the whole mRNA would code for protein, but in fact it doesn't. The protein coding sequence of an mRNA is only a portion of the whole mRNA. Remember our example mature, fully processed mRNA from the transcription video? We saw that mRNA get a 5' cap added on, as well as a 3' poly-A tail, and 4 exons spliced together, removing 3 introns. The part of the mRNA that will be read to build a protein is only this portion indicated in red. The nucleotides upstream toward the 5' end and downstream toward the 3' end don't code for protein and are not translated. These regions are called the 5' and 3' untranslated regions, or UTRs. Although they don't code for protein, they may have important regulatory functions, like serving as binding sites for RNA binding proteins that will control how long this particular mRNA will stick around in a cell, or where in the cell this mRNA might be transported to. The start codon AUG marks the beginning of the protein coding segment, and the stop codon signifies the end. These are the molecular signals that tell the ribosome where to begin and end the translation process. The other thing to recognize is that this will set the correct reading frame for translation. The ribosome will begin at AUG and translate the following codons in sequence from that point on. So if this were an mRNA being translated, the ribosome would begin here. AUG specifies methionine. The next codon is GAC, which specifies aspartate. The next codon is UGU, which specifies cysteine. The next codon is CGA, which specifies arginine. And then the stop codon signals termination. There's no amino acid. Protein synthesis is complete. So the start codon signals where to start and sets the frame in which the codons will be read. Finally, the genetic code is nearly universal. With few exceptions, the codons are the same across all organisms. This is, of course, due to our common ancestry. The genetic code evolved early and has been passed on mostly unchanged, remaining nearly the same across phyla. One consequence of this is that gene sequences can be transferred from one species to another, and will still be transcribed and then translated appropriately. This happens all the time in nature. Various vectors, viruses for example, can pick up bits of DNA from one species and transduce them into another species. And we have lots and lots of evidence for this type of horizontal gene transfer between species throughout evolutionary history. But it's also the basis for more recent technological genetic modification. An example is the creation of Bt crops with genetic pest resistance. Crop loss due to insect damage is a huge problem in farming, which conventional farmers traditionally have controlled using synthetic or naturally occurring pesticides. There are economic costs associated with this, as well as potential health risks associated with consumption of these pesticides on food. While the plants have their pests, pathogens, and parasites, the pests themselves have their own. Remember that arms race we've talked about several times in the class? Many species, especially pathogens, produce proteins and other compounds that are toxic to other species. Bacillus thuringiensis, Bt, is a soil-dwelling microbe that's also a parasite of the larval stages of various butterfly and moth species, like corn borers and other agriculturally important pests. They produce a toxic protein called cry, short for crystalline, not because it makes insects cry, although maybe it does. Farmers have been using Bt bacterial spores or the purified toxic cry protein as a natural insecticide for decades. Once technology became available to genetically transform plants, researchers set to work to cut out the middleman, as it were. Scientists have taken the cry gene from these bacteria and used genetic tricks to get that gene into economically important crops like corn and cotton. Expression of the gene and translation of the protein in the plant tissue makes the plant resistant to those insect pests. Essentially, the plants are now genetically modified to produce their own natural pesticide. The plants have no issue translating the bacterial cry protein because the genetic code is universal. That's not to say there are no problems with this approach to pest control. That could be the topic of a whole separate discussion, but that's not the focus of this video. We're here to talk about translation. So we know that the code for building the protein is contained within the sequence of nucleotides in the mRNA, which itself is copied from the protein-coding gene in the genome. Before we get into the details of the mechanism of how that protein gets built, I want to talk about two other important players in this process: the ribosome, which is the factory floor where protein synthesis takes place, and the tRNAs, which we can think of as the delivery trucks that deliver the correct amino acid at the correct time during synthesis of the protein. The ribosome consists of two subunits, a large subunit and a small subunit that sandwich the mRNA between them. You can see the mRNA down here at the bottom. Here's the 5' end, and here's the 3' end. This mRNA is then fed through the ribosome in the 5' to 3' direction and read one codon at a time, and the appropriate corresponding amino acid is added to the protein being synthesized at that position. As I said, the tRNAs are responsible for delivering the amino acids, and the inside of the ribosome has 3 sites through which the tRNAs will pass as they deliver their amino acids: the A site, the P site, and the E site. The A site is where all incoming tRNAs, after the first one, will enter the ribosome, bringing the next amino acid to be added to the chain. A stands for aminoacyl. An aminoacyl-tRNA is one that has been enzymatically linked to its amino acid cargo. tRNAs next transition through the P site as the mRNA moves through the ribosome. P stands for peptidyl, and the tRNA in this site is covalently connected to the growing polypeptide chain. Finally, tRNAs transition to the E site. E stands for exit, and empty tRNAs no longer connected to amino acids move through this site after delivering their payload. As we've talked about previously in the class, ribosomes are made up of both protein and ribosomal RNA, or rRNA. And this next illustration of the ribosome really makes clear how significant the rRNA components of the ribosome are. In purple are the protein components of the large and small subunits, and in pink are the rRNA molecules. You can see the helical structures formed by intramolecular hydrogen bonding in each rRNA molecule. rRNAs are a class of functional RNA molecules. They don't code for protein like mRNA. They carry out their function in the ribosome as RNA molecules. This animated view shows you all sides of the ribosome, and you can see that the rRNA components of each subunit are significant. These rRNAs are crucial for the function of the ribosome. One of the small subunit rRNAs is critical for matching up to and finding the correct place in the mRNA to start translating. And one of the rRNAs in the large subunit is the enzymatic component of the complex. This is a ribozyme, an RNA with catalytic activity. In this case, the catalytic activity is responsible for the peptide bond formation between amino acids. Now let's take a closer look at the tRNAs. Transfer RNAs, or tRNAs, are also functional RNA molecules. And the function of tRNA in translation, as I said, is to deliver the correct amino acid at the correct position as the polypeptide chain is being built. They are the bridge between the language of nucleotides and the language of amino acids. The decoder ring, if you will. Younger viewers might have to Google that, I know. Like all RNA, they are single-stranded, and once transcribed, they fold up into a characteristic cloverleaf secondary structures like you see here due to intramolecular hydrogen bonding between complementary base pairs that form when the single-stranded molecule folds back on itself. You can trace the single tRNA molecule from the 5' end here all the way around these stem loops to the 3' end here. Different regions of the tRNA molecule then interact via additional hydrogen bonds to form the tertiary structure you see in the space-filling model of tRNA on the left. Functionally important regions of the tRNA molecule include the anticodon down here at the bottom, which will match up with the corresponding codon sequence in mRNA during translation, and the acceptor stem at the 3' end, which is where the correct amino acid is enzymatically attached to this tRNA molecule. During translation, only the tRNA that has the complementary anticodon and is therefore carrying the correct amino acid that matches up via hydrogen bonding to the codon being translated in mRNA will be allowed to enter the ribosome. For example, this tRNA has an anticodon with the sequence AAA, and it has been enzymatically covalently bonded with its correct amino acid, which is phenylalanine. This tRNA will only be allowed to enter the ribosome when the mRNA codon sequence UUU enters the ribosome. The codon UUU specifies the amino acid phenylalanine and essentially recruits the correct tRNA carrying that amino acid via the hydrogen bonding interaction between the UUU codon and the AAA anticodon. tRNAs with other anticodon sequences holding other amino acids will be rejected and not allowed to enter the ribosome. You might predict that since there are 61 codons in the standard genetic code, there must be 61 tRNAs to match up with those codons, but it turns out that cells have far fewer, because there's some promiscuity allowed with the anticodon-codon pairing. In other words, a single tRNA can potentially match up with more than one codon, but only those codons that specify whatever amino acid it's carrying. This is due to structural flexibility in the 5' nucleotide position of the anticodon. The nucleotide here can shift its position slightly to pair up with non-standard partners. For example, G can pair with U at this position. This is referred to as wobble. How does a particular tRNA end up being connected to the correct amino acid? Obviously, that's not going to be random. If the tRNA with the anticodon AAA is supposed to be connected to the amino acid phenylalanine, something has to ensure that that's what happens. As is typical in cells, this process is catalyzed by an enzyme, an aminoacyl-tRNA synthetase, which creates a covalent bond between amino acids and the tRNAs they correspond with. The synthetase enzyme is depicted here in green, and in yellow is the amino acid this enzyme links to tRNA, and the tRNA is shown in red. Any given synthetase enzyme will recognize its amino acid and all of the tRNAs that should get linked to that amino acid. Remember, redundancy means there's usually more than one codon that specifies an amino acid, and so there might be more than one tRNA that should get linked to that amino acid, depending on wobble. So how many aminoacyl-tRNA synthetase enzymes are needed in the cell? 20, one for each amino acid. How does the enzyme know which tRNAs to link that amino acid to, since there could be 30 or 40 different types of tRNA in the cell? As you can imagine, it's the anticodon loop sequence that is the determining factor here. The enzyme feels out the nucleotide sequence in the anticodon loop to identify the correct tRNA partners for its amino acid before making that covalent connection between them. Once that connection is made, this aminoacyl-tRNA is released and joins the pool of tRNAs in the cell just waiting their turn to participate in protein synthesis. Now that you've been introduced to the major players, let's see how they all work together in the context of translation. Translation is divided into 3 stages: initiation, elongation, and termination. We're going to look at this process as it occurs in eukaryotes, which is a little bit different than what happens in prokaryotic cells. The initiation of translation in eukaryotes is facilitated by a set of proteins called initiation factors. These proteins, along with the first tRNA met, the one that binds to the start codon and is carrying the amino acid methionine, bind to the small ribosomal subunit and guide it to the 5' cap of an mRNA, where the whole complex binds. Next, we see translocation of this complex down the mRNA toward the 3' end until the start codon is reached. This is referred to as 5' to 3' scanning. Now, AUG is used as the start codon, but it's not the only sequence that's important for this start site recognition, and that makes sense. You could have many AUG sequences in a given mRNA, in the protein coding regions and in the 5' untranslated region as well. The probability of running into these 3 nucleotides in sequence is 4 times 4 times 4, or every 64 nucleotides. And we're talking about mRNAs that are thousands of nucleotides long usually. So the ribosome relies not just on the AUG, but also some surrounding nucleotide sequence called the Kozak sequence, after Marilyn Kozak, the scientist who identified it. A good Kozak sequence looks like this: GCC, ACC, AUG, G. Once the small subunit and tRNA and initiation factors encounter this good Kozak sequence, the complex pauses, positioning the small ribosomal subunit such that the AUG sequence is positioned at the P site in the ribosome, with the initiator tRNA met in the same site. Next, the large ribosomal subunit joins the complex and the initiation factors dissociate, leaving the assembled ribosome, mRNA, and initiator tRNA met in position and ready to begin. Every stage of protein synthesis is an energy-intensive process. This initiation process costs energy in the form of ATP, which is required for 5' to 3' scanning, and GTP, which is essentially an ATP equivalent, which is required for final assembly of the initiation complex. Now we're ready for elongation. With the initiator tRNA met in the P site, the A site next door is open and ready to accept the next tRNA and start protein synthesis. Which tRNA enters is determined by the codon sequence in the mRNA that is positioned in the A site. For example, if this codon sequence in the A site is CCC, then the only tRNA that can enter and bind is the one with the corresponding GGG anticodon. And as we know from the genetic code table, the codon CCC specifies proline, so that's the amino acid the entering tRNA would be covalently linked to. Just as we saw with initiation, elongation requires the participation of additional proteins called elongation factors. An elongation factor is responsible for chaperoning the correct tRNA to the A site, and dropping off that tRNA is an energy-requiring process. 1 GTP is used per tRNA. Once the 2 amino acids on their tRNAs are positioned next to one another in the P and A sites, the enzymatic activity of a large subunit rRNA, the ribozyme, catalyzes peptide bond formation between the amino acids, which breaks the covalent bond between the amino acid in the P site and its tRNA. So essentially, the tRNA in the A site is now holding a dipeptide consisting of the amino acid it's linked to and the first amino acid, now connected together by a peptide bond. A different elongation factor comes into the ribosome near the A site and ratchets the whole complex over exactly 3 nucleotides, such that the A site tRNA with its dipeptide is now sitting in the P site. The tRNA that was in the P site moves to the E site and exits, and the A site is again open for the next tRNA to enter the complex, bringing the next amino acid. This translocation also requires 1 GTP to be hydrolyzed, so every amino acid added to a protein being synthesized requires 2 GTPs to be hydrolyzed, one for dropping the tRNA in the A site, and the other for translocation of the mRNA through the ribosome. Then the cycle just repeats again and again till a stop codon enters the A site. When a stop codon enters the A site, another set of proteins becomes involved, the release factors. No tRNA enters the A site when a stop codon is positioned there. Instead, a protein release factor positions itself in the A site and catalyzes hydrolysis of the polypeptide from the tRNA positioned in the P site, releasing the newly synthesized protein from the ribosome. Then, when elongation factor binds to the ribosome and tries to ratchet the mRNA through, The release factor bound in the A site functions like a crowbar and pries the two ribosomal subunits apart, dissociating the ribosome from the mRNA. There are several release factors that are involved in this process. We won't go through their individual roles, we'll just look at their general role. Now that we've seen how the process of translation works, I want to show you a lovely animation of the process that shows an accurate structural representation of the ribosome, tRNAs, elongation factors, and release factors, and the processes of elongation and termination. This animation was produced in the lab of Venki Ramakrishnan at Cambridge University in the UK. In 2009, Ramakrishnan shared the Nobel Prize in Chemistry for his work on the structure of the ribosome. This video shows elongation and termination in a bacterial system, so the names of the proteins are different than in eukaryotes, but the mechanism is pretty much the same. Here's the large ribosomal subunit up here, the small subunit down here, here's the mRNA being threaded through, and the initiator tRNA positioned in the P site. The A site is empty. On the right side, we see an elongation factor bringing the next tRNA and its amino acid cargo. If the anticodon matches, then the elongation factor releases the tRNA into the A site. In other words, if it fits, it sits. That little flash was meant to represent GTP hydrolysis, which is required for the tRNA to be released. The large subunit rRNA right here catalyzes peptide bond formation, and now this tRNA is holding the dipeptide. A different elongation factor ratchets the mRNA through exactly 3 nucleotides, moving these two tRNAs to the E and P sites. Again, there's GTP hydrolysis required for this, and the A site is now empty. The next tRNA is brought to the complex and dropped into the A site. You can see the first tRNA exiting the E site at the back. And the process continues until a stop codon is reached in the mRNA. Compared to DNA replication and transcription, which can see the polymerization of nucleotides at up to 1,000 residues per second, translation is relatively slow. In eukaryotes, fewer than 10 amino acids per second are added to the polypeptide. Largely, that's because different tRNAs are competing to enter the A site, so it takes longer for the right one to make it in. You can see the polypeptide emerging out the top of the large subunit here. Once the stop codon is reached, release factor enters the A site and catalyzes hydrolysis of the peptide from the tRNA in the P site, releasing it. Another release factor takes the place of the first, and when elongation factor tries to ratchet the mRNA through, the whole complex dissociates and translation terminates. I love this animation. It makes the process so much easier to visualize. I've given you the link to the whole video in the PowerPoint slides if you want to watch the whole thing. Now that we've delved into the details of how a sequence of nucleotides specifies a particular sequence of amino acids in a protein, we can return to the topic of mutation and look at the consequences of changes to DNA sequence a bit more closely. First, let's look at single base pair substitutions, where one nucleotide base pair has been changed to another. We've talked about these single nucleotide polymorphisms, or SNPs, before in the class. This could be the result of a mismatch during replication that wasn't caught and repaired and ended up changing the sequence of DNA at a single base pair. Say, an AT base pair was converted to a GC base pair. This type of substitution can happen outside of coding regions in the genome, outside of genes. Since only about a quarter of the human genome, for example, is made up of genes, the rest is repetitive DNA and other stuff that doesn't code for proteins. It's more likely a mutation will occur outside genes than within them. So if that happens, typically no harm, no foul. Even within genes, we might have a base pair substitution in a non-coding region. Remember those introns we talked about in the transcription video? Unless a splice site sequence is being disrupted, that probably won't have any effect on the protein either. But let's say that the base pair change happens in a gene and in the coding region, an exon of a gene. What would be the consequence? Well, as usual in biology, it depends. It could be what's called a synonymous substitution. Because the genetic code is redundant, you can change the nucleotide sequence of a codon and not change the amino acid encoded. For example, let's return to our example mRNA sequence we saw before, which we translated to yield the sequence MET, ASP, cis, ARG. I've left off the 5' cap and poly A tail just for simplicity's sake. If there was a mutation in the gene that made it so that this codon were UGC instead of UGU, that wouldn't have any effect on the protein at all, since both UGU and UGC encode the same amino acid, cysteine. So that's what's meant by synonymous substitution, no change to the amino acid. A non-synonymous substitution changes the codon to one that codes for either a different amino acid, which is called a missense mutation, mis meaning wrong, it's encoding the wrong amino acid, or to a stop codon, which is called a nonsense mutation. Non meaning lack of, it doesn't code for anything. For example, if the UGU codon were changed to UGG, that now encodes tryptophan. So during translation, instead of cysteine being incorporated into the protein at this position, a tryptophan would be inserted instead. How harmful would that be? Again, it depends. Is that a conservative substitution or non-conservative? Do cysteine and tryptophan have the same properties in terms of polarity and charge, or are they different? How important is this amino acid position to the structure and function of the protein? Both of these considerations will determine how harmful this missense mutation is. If the UGU codon were changed to UGA, that would introduce a stop codon at this position, and protein synthesis would terminate prematurely. How harmful this sort of premature truncation is depends on where it occurs in the protein. This example protein is just a small peptide consisting of only 4 amino acids, but remember the average protein is around 300 amino acids in length. What if a premature stop codon were introduced in the second to last, in other words, the 299th codon in the chain? Would missing those last 2 amino acids have a significant effect on the protein? Maybe, maybe not. What if it happened in the third codon out of 300? Well, that's a complete loss of the protein, so that would be extremely detrimental. What about other types of mutations? What if we're talking about insertions and deletions rather than single nucleotide substitutions or polymorphisms? Insertions and deletions, or indels, are generally much more problematic than SNPs because they usually shift the reading frame of the mRNA. Remember that translation begins at AUG and then proceeds through the following triplets sequentially. Insertion or deletion of one or more nucleotides can significantly change the identity of the codons that follow by disrupting the reading frame. Let's look at some examples. Let's say that a mutation occurs at the gene encoding this protein such that now there's an extra uracil, a U immediately following the AUG start codon. What would be the impact of that insertion? The ribosome would begin translating at the start codon, the AUG, then when the next codon is decoded, what is it? It's no longer GAC, now it's UGA, and that's a stop codon. So the effect of this mutation would be immediate nonsense and premature termination of translation. What if the extra U is after the next G rather than before it? Well, now when the mRNA moves through the ribosome, all of the codons following the AUG are different. The entire amino acid sequence of the protein has been changed. So the gene is now encoding a completely different protein. What would this protein do? I have no idea, but I do know that it's not going to have its normal function. I'll leave you with this final example. What would happen if instead of a single nucleotide insertion, what if there were an insertion of 3 nucleotides, resulting in 3 extra uracils in the mRNA at this position? What would be the consequence of that to the encoded protein? I'll leave you to figure that out. We'll go over that and some other examples in class.